Module 9 UHS Modular Integrated Undergraduate Curriculum Physiology, Dr. Sheen, CEO and founder, Sheikh Zayed Medical College Physiology Department. Medical Superfast Simplified Physiology Video Lecture Notes. Guyton and Hall 14 edition, chapter number 51, lecture number 13, page 642, 643. Learning objectives. Describe the mechanism of excitation of rods, receptor potential. Mechanism of excitation of rods. Excitation of the rod when rhodopsin is activated by light. The outer segment of the rod, where the photoreceptor discs are located, has the rod membrane. In the dark state, is leaky to sodium ions that flow through cGMP-gated channels. In the dark state, cGMP levels are high, permitting positively charged sodium ions to continually leak back to the inside of the rod and thereby neutralize much of the negativity on the inside of the entire cell. Thus, under normal dark conditions, when the rod is not excited, there is reduced electronegativity inside the membrane of the rod, measuring about -40 millivolts. When light hits the photoreceptor, for example, rod cell, the light-absorbing retinal portion of rhodopsin is activated. This stimulates transducin, a G protein, which then activates cGMP phosphodiesterase. This enzyme catalyzes the degradation of cGMP into 5-GMP. The reduction in cGMP then causes closure of the sodium channels, which, in turn, causes hyperpolarization of the photoreceptor. Then, when the rhodopsin in the outer segment of the rod is exposed to light, it is activated and begins to decompose. The cGMP-gated sodium channels are closed, and the outer segment membrane conductance of sodium to the interior of the rod is reduced by a three-step process. 1. Light is absorbed by the rhodopsin, causing photoactivation of the electrons in the retinal portion. 2. The activated rhodopsin stimulates a G protein called transducin, which then activates cGMP phosphodiesterase. This enzyme catalyzes the breakdown of cGMP to 5-cGMP. 3. The reduction in cGMP closes the cGMP-gated sodium channels and reduces the inward sodium current. The excitation cascade mechanism by which rhodopsin decomposition decreases membrane sodium conductance. Only 30 photons of light will cause half saturation of the rod. How can such a small amount of light cause such great excitation? The answer is that the photoreceptors have an extremely sensitive chemical cascade that amplifies the stimulatory effects about 1,000,000-fold, as follows. 1. The photon activates an electron in the 11-cis retinal portion of the rhodopsin. This leads to the formation of metahodopsin 2, which is the active form of rhodopsin. 2. The activated rhodopsin functions as an enzyme to activate many molecules of transducin, a protein present in an inactive form in the membranes of the discs and cell membrane of the rod. 3. The activated transducin activates many more molecules of phosphodiesterase. 4. Activated phosphodiesterase is another enzyme. It immediately hydrolyzes many molecules of cyclic guanosine monophosphate cGMP, thus destroying it. 5. Within about a second, another enzyme, rhodopsin kinase, which is always present in the rod, inactivates the activated rhodopsin, the metahodopsin 2, and the entire cascade reverses back to the normal state with open sodium channels. Thus, the rods have developed an important chemical cascade that amplifies the effect of a single photon of light to cause movement of millions of sodium ions. This explains the extreme sensitivity of the rods under dark conditions. The cones are about 30 to 300 times less sensitive than the rods, but even this allows color vision at any intensity of light greater than extremely dim twilight. Rod's receptor potential. The rod receptor potential is the electrical response of rod photoreceptor cells in the retina to light stimulation. This response is critical for initiating the process of vision, particularly in low light conditions. The receptor potential in rods is unique because it involves hyperpolarization rather than the depolarization seen in most other types of neurons. Characteristics of the rod receptor potential. Hyperpolarization. Unlike most neurons, rods hyperpolarize in response to a stimulus light. The receptor potential becomes more negative as light intensity increases. The receptor potential in rods is graded, meaning that its magnitude varies depending on the intensity of the light. Stronger light causes a greater reduction in cGMP, leading to more significant channel closure and more hyperpolarization. Rods have a slower response to changes in light compared to cones. That is why they are better for low vision rather than detecting rapid changes or bright light. Conclusion: the rod receptor potential is a hyperpolarizing response to light that reduces the release of glutamate, thereby changing the signal transmitted to the bipolar cells. This process is essential for night vision and allows the visual system to detect and process low levels of light.